Hi everybody. Going to try to attempt to use this program to do an online PowerPoint, so hopefully this goes well. The topic that I'm going to be talking about today is nucleotides and nucleic acids, and specifically be talking about DNA and RNA. And so this presentation is going to focus on the DNA, and then the one I will post on Wednesday will focus on RNA mostly. So I'm going to start talking off about the structure of DNA. Just a couple of reminders and a little bit more details of the structure, and then start on DNA replication and DNA repair. So DNA and RNA are made up of the monomer of nucleotides. So I'm just going to talk a little bit about the terminology. So if you look here, we have what is a nucleotide, which includes the nitrogenous base portion, so the base portion here, which can either be a purine or a pyrimidine, and I'll go into some more details about the purine and the pyrimidine. The sugar portion, the pentose sugar portion here in the middle, and depending on whether it's DNA or RNA, we'll have either the hydroxyl group on the 2' carbon, which is the nucleotides that are part of RNA, or without the hydroxyl group, which are the nucleotides that are part of DNA. And then we have the phosphate group that is attached to the 5' carbon here on the sugar. Okay, so that makes up a nucleotide. A nucleoside includes only the nitrogenous base and the pentose portion of the molecule. So we're going to look into each portion of the nucleotides and talk a little bit about each one of the portions. So first we're going to focus on the phosphate group and the different characteristic of the phosphate group. And again, I want you to notice that that phosphate is again attached to that 5' carbon that's located on the pentose sugar. So the phosphate group on the nucleotide is negatively charged when we are looking at it at neutral pH. So the oxygens on that phosphate group are negatively charged, typically attached to the 5' portion, so the 5' carbon that I just talked about. Nucleic acids, when we're talking about building nucleic acids, so this is including both DNA and RNA, they're built using the triphosphate version of the nucleotide. So those are ATP, GTP, TTP, and CTP. And that will include the sugar, the base, and then 3 of the phosphates. And the pictures that I've shown you here have that 2' carbon, so these would be the ones that would be incorporated into RNA. But again, we're looking at the triphosphate where it has 3 phosphates attached to the nucleotide. And those are used in building DNA and RNA. So when you're building the nucleic acids, 2 of the 3 phosphates form a leaving group, and the complete nucleic acid contains only 1 of those phosphates. So when we look more closely at DNA and RNA, you can see that the bonds that link the nucleotides together are phosphodiester bonds. So they just contain 1 of those phosphate groups. Okay, so now I'm going to talk a little bit about the sugar portion of the nucleotide. So the sugar portion of the nucleotide is made up of a pentose sugar, and the form of that pentose sugar differs from some nucleic acids and nucleotides. So in RNA, we find the beta D ribofuranose, and this is a picture of the fructose right here, where in DNA we find the beta 2' deoxy D ribofuranose. So again, that deoxy portion not having the hydroxyl group at that 2' carbon. So the sugar ring actually is able to take on a couple of different conformations, and we call these different conformations puckered conformations in that sugar ring. So here is a picture of the 2 different puckers of the sugar, and you see when we're looking at the structure, depending on the location of the base, we have a different formation of that ring structure, and that will give a different angle between these oxygens here. So now let's focus on the purine and pyrimidine, or the nitrogenous base part of the molecule. So down below here you have a generic example of a pyrimidine and a purine, and you can notice off the bat the difference between the two. So the difference between the single ring structure as opposed to the double ring structure seen here. So the nitrogenous bases that we'll look at, and I'll show you examples of each one of the nitrogenous bases we see in both DNA and RNA. They're nitrogen-containing heteroaromatic molecules, meaning that they contain both carbon and nitrogen in the aromatic rings. The structure of the pyrimidines and purines is planar or almost planar, so it's almost a flat structure. And we can identify them by their absorption of UV light around 250 to 270 nanometers. So first we'll be looking at the purines, and we have two purines, the adenine and the guanine. And we can see that they both have the double ring structure containing both carbon and nitrogen. Obviously, I'm not very good at drawing with this pen. Anyway, all are good at hydrogen bonding, and we'll look at the hydrogen bonding between these purines. And these molecules are both neutral at pH 7, so they do not have a charge on them at pH 7. The pyrimidines include cytosine, which is found in DNA, thymine, which is also found in DNA, and then uracil that replaces thymine in the RNA. And you can see they all have the very similar structures, all able to form hydrogen bonds, and I will show you the hydrogen bonding between the molecules that are used in both DNA and RNA. And again, as I mentioned, you're going to have to memorize the structures of each one of these and memorize the hydrogen bonding between the two that occurs. So in nucleotides, the pentose ring, so the sugar ring, is attached to the nitrogenous base via an N-glycosidic bond. Okay, so this is where the base attaches to the sugar. So the bond is formed at the one of the carbons of the sugar in a beta conformation, and I'll show you the structure of that in a second. And the bond is formed between the N1 on pyrimidines and on the N9 of purines. Okay, so that is bonded to the carbon on the sugar. This bond is quite stable, especially in the pyrimidines, so it's very infrequently able to be hydrolyzed. So again, we're talking about the bond here between this carbon on the sugar and the nitrogen on the nitrogenous base, depending on if you're a purine or a pyrimidine. So that bond, that glycosidic bond right there, oh, I'm still not good at drawing this, has free rotation. So it allows the base to rotate in either direction. Okay, so it can be on the same side as the sugar here or on the opposite, so in the syn side or in the anti side of the sugar. When we call the angle that's at the 0, that's the syn conformation, and when we're 180, that's the anti. So this one would be the anti here with the sugar, the base on this side of the sugar, and the syn would be with the base on this side of the sugar here. Okay, so those were the nucleotides. Let's talk about the idea of polynucleotides, so joining nucleotides together. So that's our formation of DNA and RNA. So covalent bonds are formed between the nucleotides, and in the case of DNA and RNA, we call these phosphodiester linkages. those phosphodiester linkages, as you remember, as I told you, the phosphates are negatively charged. So these phosphodiester linkages give the backbone of RNA and DNA a negative charge. So for those of you who've taken genetics or have run DNA gels, the DNA is able to run towards the negative to the positive because it is negatively charged. So these are linear polymers. There are no branching or cross links like we see in the glycogen. There's no branching at all. It's a linear polymer of the nucleotides. The strand has what we call directionality, meaning that the 5' end and the 3' end are different. And when we read the sequence of DNA, we always read it from the 5' to the 3' end. So if we're looking at the A's, G's, and C's, and T's, we're reading that from the 5' end to the 3' end of the molecule. The backbone of DNA is fairly stable. It is very hard to break down DNA, and we'll talk about the situations where DNA backbone can be broken. RNA, on the other hand, is far less stable. In water, RNA lasts only for a few years. In cells, mRNA can be degraded in a few hours. And that comes from that 2' hydroxyl, and we'll talk a little bit about how that 2' hydroxyl makes RNA less stable in this talk on Wednesday. Okay, so here's a picture of the phosphodiester bonds. So we have the nucleotide adenine here being linked to the next nucleotide here through what we call a phospho, because it has the phosphate, and then a diester bond linking these two nucleotides together. And we have the negatively charged backbone. Same is true in RNA. We have our one nucleotide attached to the next nucleotide through the phosphodiester bond here. So 2 oxygens, a phosphate, phosphodiester bond. Okay, and then we'll talk about the sequence of adding these nucleotides together. Specifically, we'll focus on DNA today, so DNA replication, and then RNA on Wednesday. Here is located our 5' end, so the 5' end contains the phosphate, and here is located our 3' end, and our 3' end contains the hydroxyl. So let's talk about the hydrogen bonding interactions that occurs between the bases. So 2 bases can form hydrogen bonding base pairs. So for monomers, so just a single monomer of a nucleotide, there's a large number of base pairs that are possible. But in polynucleotides, so DNA and RNA, when they're linked together, there are very few possibilities that exist. So Watson and Crick base pairs, so the Watson and Crick idea of base pairs predominantly in double-stranded DNA states that A's always pair with T's and C's always pair with G's. Okay, so purines pair with pyrimidines. So if we look more closely at that, so if we have our double-stranded DNA, so we have our 5' end here that has our phosphate group, our 3' end that has our hydroxyl, and then the other strand that goes anti-parallel has the 5' phosphate and the 3' hydroxyl. Okay, so we're talking about base pairing that occurs here between the 2 nitrogenous bases of the opposing DNA. Okay, so if you look a little bit closer at that, you can see where the base pairs occur between adenine and thymine. We have 2 base pairs, so A and T has 2 base pairs, whereas guanine and cytosine has 3. Yes, you need to memorize where these base pairs are located. You will be drawing these structures on the final exam. So because the AT base pairing only has the 2 hydrogen bonding, we consider that a weaker hydrogen bond pair than the G and C. So organisms like bacteria that live at really high temperatures tend to have genomes that are high GC, stabilizing their genomes, stabilizing the interactions between their DNA. So let's look a little bit at the model of the structure of DNA, and this was the model derived by Watson and Crick using Rosalind Franklin's data to determine the alpha helix structure. So we have here the backbone, which is the blue ribbon here. Here it would be the blue representing the sugars here. And then on this model, this would be here representing the sugars. On the inside, we have represented here the base pairing, which is represented in the middle here. And you can see from this picture here how that structure of the nitrogenous bases is in that planar flat structure there. And then here, the space filling model, you can see the bases on top of each other here. Okay, so we've got the double strands running anti-parallel, forming the alpha helix with the base pairs occurring in the middle of the molecule. We have two grooves in the DNA, what we consider the major groove and the minor groove. And this actually allows for interaction of DNA binding proteins in different portions of the molecule. And because the major groove is bigger than the minor groove, we tend to have most of our protein interactions in that major groove. This is just another picture showing that similar interaction. Again, we have our anti-parallel strands, 5' to 3', 3' to 5', our base pairing occurring in the middle, and then a space filling model. And this one shows the specific molecules. So the red being the oxygen, the carbon being the blue, and then the carbon and the nitrogen and the base being this orange color here in the middle. And then the yellow being the phosphate group. So you can see phosphate group here, phosphate group there. Okay, now that we've reviewed a little bit of the structure, I'm going to talk about DNA metabolism. So what is DNA metabolism? So DNA provides for the cell a stable storage of the genetic information, but the structure of DNA is far from static. It's not always in a static structure. So every time we need to copy the DNA or to divide a cell, we need to synthesize a new strand of DNA, and that synthesis has to occur with high fidelity, meaning that DNA synthesis cannot have very many mistakes. So before each cell division, an organism needs to copy its entire DNA, and we refer to that as DNA replication. Errors arise during DNA synthesis, so the DNA constantly needs to be checked, as well as any sort of damage that can be done to it, and we refer to this as DNA repair. And then finally, segments of DNA can be rearranged with a chromosome or between any DNA molecules, giving a new novel piece of DNA from that interaction, and we call that process recombination. So the process of DNA metabolism includes replication, DNA repair, and recombination. And we're going to focus on mostly today DNA replication and a little bit on DNA repair. So most of you guys have been taught DNA replication, maybe by me, maybe by someone else, a couple of times in your careers. So we're going to just do a brief overview of the process of DNA replication. So for DNA to be replicated, it needs to make a copy of itself. Strand first has to separate. So we can see we have the division of the original alpha helix, which is here in blue, into single-stranded molecules. That single-stranded molecule serves as a template for the new strand, which is represented here in pink, to make a copy of that DNA. DNA is replicated by enzymes we call DNA polymerases. So polymerases are responsible for replicating DNA. So a newly made DNA molecule has one strand of it that's new. So that in this case would be the pink strand, and one strand that is old. And we call that semi-conservative DNA replication. Okay, so what is happening in DNA replication? Well, it is the process of linking two nucleotides together via that phosphodiester bond. So the phosphate group is being attached at the 3' hydroxyl. And in that process, we lose the water. So ultimately, we're doing, again, creating that phosphodiester bond between two nucleotides. And we're going to go into detail about how that process happens. So there are three fundamental rules of replication. The first one being what we just talked about, which is replication is semi-conservative. So in DNA replication, one of the strands is from the old DNA, and then one is a new strand. Okay. Replication begins at a certain point. We call that the origin of replication, and it proceeds biodirectionally, meaning replication happens from the origin and then goes out from that origin in both directions. Synthesis of a new DNA molecule occurs in the 5' to 3' direction. And it is also what we call semi-discontinuous. So it doesn't happen always in a continuous form, and we'll talk about why that happens as well. So we're going to talk about DNA replication in E. coli. And for those of you who haven't studied a lot about E. coli, the chromosome of E. coli is circular. So it is a double-stranded circular chromosome. Doesn't that look so pretty? So the DNA is different from what we have in humans, eukaryotes, which is linear chromosomes. So the DNA in bacteria is circular. So they need to replicate this circular piece of DNA. The replication starts at what we call the origin of replication. We'll talk about that a little bit more closely in a second. And we create two daughter duplicates of that original circular DNA. Okay, so the origin of replication. So the DNA replication's got to start somewhere. In bacteria, it happens at a region that is rich in A and T. Okay. Again, as you remember, the A and T hydrogen bond only has 2, which makes it a little less stable at that point. At that point, the DNA can bubble open. So at that double strand, you get what we call a bubble. Oh, my pictures are so bad. I apologize. So it bubbles up at that origin of replication where DNA can start. So in E. coli, that origin of replication is called ORI-C, and it's about 245 base pairs in length. It's a highly conserved sequence among bacteria. It repeats 5 base pair sequence, which we call the R site, and forms a binding site for the protein called DNA-A, which initiates DNA replication in E. coli. Again, it's an AT-rich region, and that allows for the DNA to be unwound at that portion. Additional sites include the I site where DNA-IA binds, the IHF, or the integrated host factor, and the FIS, which is the factor for inversion stimulation. So DNA synthesis proceeds in the 5' to 3' direction. So synthesis always occurs by adding new nucleotides to the 3' end of the growing molecule. In DNA replication, we have 2 strands, and I'll show you a picture of it in a second, that make up the replication of fork, one of them being the leading strand that's made continuously, and the other one being the lagging strand that's made discontinuously. In other words, it's made in short pieces that we call Okazaki fragments, after Mr. Okazaki, that are later joined together to make a continuous piece of DNA. So here again is the replication fork. So remember, we had our origin of replication, and the DNA began to bubble up at that point. So it became to be unwind and form two single-stranded portions here. Okay, so this is where DNA replications will start, and this is where we call our replication fork. So each side has one replication fork, and this picture below is just showing, for example, this side of the replication fork. Okay, so just keep that in mind that there's two sides, one going this direction and one going this direction. We're just going to focus on one side. So here we have our leading strand that is able to continuously replicate the DNA, and then the lagging strand, which is going the opposite direction. And again, this is true where we have to have the leading and lagging because we can only add new nucleotides to that 3' end. Okay, and DNA is always made in the 5' to 3' direction, so we only can add nucleotides over and over on this side. We can do that continuously, but at some point on this side, we're going to back up into the next fragment, so we got to start again and move this way, making the second strand, the lagging strand, discontinuous. And I promise when I get back next week, I'll draw some pictures of that on the board as well. So DNA is synthesized by the enzyme called DNA polymerase, or DNA polymerases, and every organism that copies its DNA has a version of DNA polymerases. So the first DNA polymerase ever discovered was in E. coli, and E. coli contains DNA polymerase 1 as well as 4 other DNA polymerases. So DNA polymerases are responsible for making the new strands of DNA. So parental DNA serves as the template for the new strand of DNA. Nucleotide triphosphates, as I talked about earlier, serve as the substrate for DNA synthesis. So in essence, we are creating a DNA molecule by adding nucleotide triphosphates to a growing strand of DNA. Okay. So the hydroxyl group at the 3' end, that's where we're adding our nucleotide triphosphate, is nucleophilic. Okay. So the incoming chain is created by the incoming trinucleotide phosphates that come in. That 3' hydroxyl is absolutely required for DNA replication to occur. Okay. So if we don't have that 3' hydroxyl, DNA polymerase can't add the new nucleotide N. Okay. So that 3' hydroxyl is also made a more powerful nucleophile by the presence of magnesium ions, and I'll show you a nice picture of that in a second. So the phosphates made by the nucleotide triphosphate, that again is the last two phosphates on the nucleotide triphosphate, are good leaving groups. So in essence, two of the phosphates are being removed from the nucleotide triphosphate, and we're forming a phosphodiester bond linked to that 3' hydroxyl. This picture here shows you the DNA polymerase and the interaction between the nucleotide triphosphate, the growing strand, and the magnesium molecule, as well as some key amino acids within DNA polymerase. So here is our template strand of our DNA going from 3' to 5', and our growing primer strand going from 3' to 5', and this is the 3' oxygen that is going to be part of the interaction that with the new nucleotide triphosphate. So the new nucleotide triphosphate comes in here, and we can see that the new nitrogenous base is forming the base pair with the template strand. So knowing that the T goes with A, it forms base pair in here, we have the correct nucleotide to come in. So we have the 3 phosphate groups. These 2 phosphate groups serve as leaving groups, and we get the formation of the phosphodiester bond. So you can see that these interactions again are stabilized by the magnesium and by these two residues on the DNA polymerase, allowing for the formation of that phosphodiester bond and the leaving of the two phosphates. So DNA polymerase, as I mentioned, requires a 3' hydroxyl for it to work correctly so that it can add the new nucleotides. So that means that it cannot start RNA or synthesis of DNA de novo. It has to have a free 3' hydroxyl to be able to start synthesizing DNA. And so the way that the cell solves this problem is by adding a short piece of RNA, an RNA primer in most cases, sometimes it can be DNA, which is a short strand that's complementary to the template. And that gives that free 3' hydroxyl so that DNA polymerase can come in there and add the next nucleotide. Most of the time, that primer is made of RNA. So let's look at the DNA polymerase in a little bit more detail. So when we're looking at the DNA polymerase, it has this almost horseshoe structure where the DNA is going through the center of that molecule. And it has these two positions in it, what we consider the post-insertion site and then the insertion site. So the post-insertion site is where the newly made base pair resides when the polymerase is moving forward, and the insertion site is where the incoming nucleotide binds. So the incoming nucleotide and where the newly made base pair has occurred. DNA polymerase can add nucleotides or disassociate. The number of nucleotides it can add before it disassociates is called processivity. The processivity of polymerases in general varies widely from a few nucleotides to many thousands. So each specific polymerase that we'll talk about has its own processivity and rate of polymerization. So you can think of it as the enzyme has to attach to the DNA. It can either attach and add nucleotides or it can disassociate from that DNA. And if it's better at attaching and making new nucleotide bonds, then it's better at adding nucleotides at a rate before it dissociates. In E. coli, as I mentioned, there are at least 5 DNA polymerases. So DNA polymerase 1, as I mentioned, was the one that was first discovered. It is abundant in the cell, but it's not ideal for replication. So its replication rate is about 600 nucleotides per minute, is slower than the observed replication fork. And it has a low processivity, so it tends to dissociate quickly from the DNA. The primary function that we found for DNA polymerase is actually cleanup, and we'll talk about that process of cleanup in a second. DNA polymerase 3 is the main DNA polymerase in E. coli, so that's the main polymerase that's involved in DNA replication. Polymerase 2, 4, and 5 are mostly involved in DNA repair. So this gives you the general breakdown of each one of those DNA polymerases in E. coli. So it has the name of the gene that codes for that, the number of subunits. So you can see some of them have more than one subunit. DNA polymerase 3 has 9, whereas DNA polymerase 1 only has 1. Some of them have proofreading ability where other ones do not. You can see that the polymerization rate differs between most of them. That DNA polymerase 3 has the highest polymerization rate. So and then it has the processivity, so the number of nucleotides you can add before the polymerase dissociates. So you can see that rate for the DNA polymerase 3, that main polymerase, is very high. In E. coli, errors in replication are very infrequent. In every 10 to the 9th, 10 to the 10th base pair, there is only about 1 error. So that's 1 error per 1,000 to 10,000 replications. So very high fidelity rate. That has to do with the base pairing between the different nucleotides that allows the correct base pair to be added most of the time. But DNA polymerases will still insert the wrong base some of the time, but there's repair mechanisms that are able to fix these errors because they do cause a conformational change in the double-stranded DNA. So we can look at the geometry of the base pairs. So when we have the correct base pairing, so the C with the G and the A with the T, we form the correct shape of that interaction. If there's an incorrect base pair, so like for example a T with a G, we have an incorrect conformation of that base pair. Okay? And that leads to the repair mechanisms coming in there and recognizing that that is a wrong base added in there. So during synthesis, errors are corrected by what we call a 3' to 5' exonuclease. Okay? All DNA polymerases have additional activity, and this 3' to 5' exonuclease is able to proofread the synthesis looking for mismatched base pairs. So the enzyme is translocated to the next position. It is inhibited until the incorrect base pair is removed and the correct nucleotide is. DNA polymerase 1 also has 5' to 3' exonuclease activity in addition to the 3' to 5' exonuclease activity, which means it moves ahead of the enzyme and hydrolyzes things in the path. It's also able to nick, creating strand breaks that move along with the enzyme. This activity and the polymerase activity are known as the Klenow fragment of the DNA polymerase. So here is DNA polymerase 1. This is the 3' to 5' exonuclease fragment there, and then the 5' to 3' exonuclease activity. Okay? So it is able to move ahead and remove nucleotides that are incorrect and replace them with new ones. So if this is an incorrect base pair located here, they can be removed and the correct DNTPs can then be added to make the correct portion of this strand. This allows for repair of mismatched bases. So DNA polymerase 3, again, that's the main DNA polymerase in E. coli, is a very complex structure with 10 different subunits. There are 2 core domains of alpha, epsilon, and theta. The core domains are linked by what we call clamp loaders. The core domains each interact with a dimer of beta subunits that increase the processivity of the complex. So the beta subunits form what we call a sliding clamp, and that sliding clamp prevents the DNA being associated from the DNA polymerase. And if I could show a video of me, I would show you what a sliding clamp exactly looks like, but you can think of it as clamping over the DNA and making sure that the enzyme doesn't disassociate from the DNA. So this makes the processivity of DNA polymerase about 500,000 base pairs because of these beta clamps that are able to clamp onto the DNA. So we've got the subunits that we consider the clamp loader that loaded onto the DNA, and then the clamp itself, the beta subunit. So if we look at the DNA polymerase, we have the core polymerase, we've got the beta sliding clamps, and then we have the clamp loader. So the core, the sliding clamps, and then the clamp loader. So if we look at each one of the subunits, we can see the different subunits here, the number of subunits per the entire enzyme, the gene, and the function of that subunit. So you'll notice a couple of things on here, the proofreading, so the looking back, the actual polymerization activity, which is the alpha subunit, the clamp opener, the clamp loader, interaction with SSB, which also refers to single stranded binding proteins. Okay. So you can see the different functions of each one, how big they are, how many are located in the holoenzyme, and what their function is. So the requirements for DNA replication to occur in E. coli requires over 20 different enzymes. So not just DNA polymerase, but a bunch of enzymes associated with the DNA polymerase. We call this the replosome. So all of the enzymes involved in the entire replication process. This includes enzymes called helicases. So helicases use ATP to unwind that double-stranded DNA. As I mentioned, those base pairs are pretty stable between the two strands, so we need energy to come in there and remove the strands. We have topoisomerases, and if you think about when we have two circular structures being replicated together, it tends to cause coil up in the DNA, and so we have enzymes that come in there and nick the DNA and are able to unwind it. We have DNA binding proteins, including those single-stranded binding proteins that I just talked about, that help stabilize those DNA strands once they are no longer in the double helix. They need to be stabilized because they really want to be together. We have the RNA primases, or the primases that are able to create those RNA primers that start the process of DNA replication. And then finally, we have DNA ligases, whose job is to seal the nicks between those Okazaki fragments together to make sure we have two continuous strands. I'm going to give you the brief overview of DNA replication and talk about what each one of those enzymes does in the process. So we have our E. coli DNA. It is single-stranded, and I drew out some pictures. Some of you guys would recognize this from genetics, to talk about what each one of these proteins does in the process. So DNA A is what we call the initiator protein. That's the one that binds to the OREC, causes that conformational change in the DNA that causes the helix to destabilize and open up. This exposes the single-stranded DNA. So we're looking at our circular chromosome of E. coli. DNA binds to OREC and is able to open up that DNA so that a portion of it is single-stranded. So we have our two single-stranded regions here in our circular chromosome. Once the DNA is open, DNA helicase, made up of what we call DNA B polypeptides, are able to assemble around that exposed single-stranded DNA, able to recruit the holoenzyme to bind to that replication fork, and subsequently keep unwinding the DNA at the two ends. Helicase requires energy supplied by ATP that is able to denature those hydrogen bonds and stabilize the double helix. The single-stranded binding proteins, they specifically bind to single-stranded DNA and they stabilize that open conformation of the helix. So DNA A was able to open up the single-stranded DNA. Helicase can continue to unwind the DNA. So you can see a DNA A is the one that breaks the seal. The helicase then can continue to break the hydrogen bonds as we go on. These single-stranded binding proteins are able to bind to the single-stranded DNA and is able to maintain that structure for the DNA polymerase to come in. So like I mentioned, there's supercoiling that occurs, and these topoisomerases include DNA gyrase, is able to relieve that supercoiling in the DNA caused by this unwinding. So DNA polymerase 1, 2, and 3 can elongate an existing DNA strand, but like I mentioned, it can't initiate DNA synthesis. It requires that free 3' hydroxyl to add the new nucleotides. So the enzyme responsible for giving them the 3' hydroxyl is the primase molecule. And primase is an RNA polymerase. It comes to the replication fork by recruitment by DNA helicase and synthesizes a short RNA primer that provides that free 3' hydroxyl for DNA polymerase to elongate. So we have our helicase, our single-stranded binding proteins, and now RNA primase can come in there and add a primer. So we have our primer, and then we begin to synthesize our new DNA from that primer off our DNA template. So the direction of the replication fork is going this direction on this side and this direction on this side. Okay. So we're just going to look at half of the replication fork and just look at half of what's happening. Again, it's happening on both sides bidirectional, but we're just going to look at one side. So let's look a little bit more closely at the DNA. So we have our DNA here. It's single-stranded on this portion, still double-stranded down here. We've made our short RNA primer that's complementary to our strand here. So the red is the RNA primer. Now DNA polymerase 3 can come in there and start adding our DNA. Again, adding it to that free 3' hydroxyl. So one of the strand we can consider continuous, the leading strand, and the second strand being the lagging strand is discontinuous. So leading would be this one that can continue, lagging strand would be the one that is in the Okazaki fragments. So we have this discontinuous synthesis where we have these small fragments. The lagging strand synthesizes those Okazaki fragments. They can come and at the end be removed by DNA polymerase, filled in by DNA, and then DNA ligase can ligate them together, giving us a full DNA. So both the strands of DNA are synthesized concurrently. And because of this, one of the strands, the lagging strand, has to be looped around, making sure that both strands are going in that 5' to 3' direction. The DNA clamp helps provide the stabilization of this interaction so that both strands can be synthesized in the correct orientation. This is a little bit more simplistic picture than what I just showed you, but it gives you that general idea of those clamps and the clamp loader and how those clamps hold on to the DNA when DNA is being synthesized. So we can see how one of the strands, the lagging strand, is being looped around in order to have both of the strands going through the DNA polymerase at the same time. And so I'm going to finish off this by showing or giving you access to a video. I'm not sure why it's not showing up right now on my thing, but hopefully you guys can play that video showing DNA replication. So with that, I'm going to end this PowerPoint for today. Hopefully you guys are able to watch it and it's fantastic and wonderful for you because I know you missed having class today. I see you guys next week. You'll hear from me again on Wednesday.